Hi, everybody. Welcome to the Chats with Chaudhrey. I hope you're safe and well. Today, I am delighted to be joined by Spanios' Dean Remy and Bhuvanesh. I hope I pronounced that correctly. And we are actually going to be talking about patient-derived tumoroids. So, gentlemen, first of all, how are you both?
We are doing well. Thank you.
Doing quite excellent. Thank you. Always good to reconnect with you as well.
Well, it's good to see you both. Right now, before we start talking about patient-derived tumoroids, though, for viewers who are not familiar with Spanios, would one of you like to give me a quick overview of the business, first of all, and what you actually do?
So Spanios is a tumoroid model development company. It was co-founded by me and our other colleagues to fulfill an unmet need in the field where they had no good model systems for rare cancer patients, which are difficult to treat.
Right. Okay. And what's your background, Bhuvanesh?
I'm a cancer biologist with around 20 years of experience, and I was a assistant professor before I started this company.
Fantastic. And Dean, what's your role and background?
Yeah, I've been in the drug discovery, sales, business development, and corporate development team building arena for about 30 plus years.
Right. Okay. Now, you said you co-founded the business, Bhuvanesh, but why did you decide to launch your own business?
So around a few years back, we had a dear friend and neighbor who developed a rare cancer. Right. And at that time, I was an assistant professor at the Houston Methodist Cancer Center, and I had a lot of collaborators all across the largest medical center in the world. So we were able to get her excellent care. But in spite of that, she did not survive beyond 9 months.
I'm sorry to hear that.
Which had a profound impact on us, more on me as a researcher, thinking, I'm here developing preclinical models for cancers for past 15 years. I see in common cancers, the response rates are very good. People survive for several years. But I found there was a huge lack of model systems to develop new targeted therapies in rare cancers. So I went about trying to solve that problem. First in the academic arena, trying to build those models and platforms in there. Then I realized this required a real focused approach. And in order to be able to do so, we decided to launch our company in May of 2017 called Spanios.
Right. Okay. And tell me a bit more about the business and in terms of the technology that you're developing.
So the goal was very simple. We built out preclinical models for where none of the other models exist. And our tech is based on an organoid-based platform, but ours are tumor-based organoids, which make it call it tumoroids. Now, for simplicity's sake, what we are trying to do is develop mechanisms by which all other drug companies can come and test their favorite drug on our model systems and allow for and facilitate easier, faster transfer of tech and development of new drugs in that manner.
Right. Now, forgive me, as a layperson, what are organoids and why are they necessary?
Organoids are basically representations of an organ. So you are trying to build something that is a mini version of whatever the original organ was. Tumoroids specifically are mini versions of the tumors. The advantages what we work on are patient-derived organoids or patient-derived tumoroids. When they're cancer-related, we are able to build in model systems that replicate what happens in a patient. And we can do testing on these without really using patients as guinea pigs. So allows us to develop and test new drug therapies and then take them to the clinic much faster, much more efficiently.
Right. And I think you've already answered this partly with your previous answer, but what makes Spanios organoids, or should I say the tumoroids, special or different or unique to what other organoids are out there?
We decided to make our organoids that were context dependent. So most organoids that are there are just the cells that cause cancer, but these grow as a mesh of multiple cells. So there is stroma in the cancer, there is immune system in the cancer, we are able to incorporate all of them while we build our model systems. And in addition to that, there's an oxygenated tumor microenvironment, which is
Yep.
extremely important. And the whole concept of building a model which is as close as possible to the human condition was our idea. And that's what we have been able to build, which is different, very different from what is available in literature.
Right. Okay. And what do your platforms enable that traditional drug testing models can't?
So half of our platforms are built on the axis of TME, which is tumor microenvironment, which allows for testing drugs that may affect the tumor microenvironment.
Right.
Additional components of that is immuno-oncology has come to the fore in the past few years. And in order to test those immuno-oncology drugs in combination with other therapeutics, you need a model system that can encompass all ends of the spectrum, which is what we are able to do. We enable all kinds of studies from small molecules all the way to immuno-oncology studies. And that is what our main advantage is.
Right. Okay. Dean, do you want to add anything to what Bhuvanesh has said so far?
Yeah, I think with a small piece or small addition, we do enable the pharmaceutical developers to test multiples of therapeutics at the same time, multiples of combinations, even preceding their developed therapeutic with checkpoint inhibitors, get a total picture of what's going on at the tumor level. But also we have the ability to run these studies much longer than all of the commercially available cancer organoid platforms that exist today. So it is more in line with how patients actually receive therapies, right? You don't just get one shot, go home and hope for the best. Often it's a series of therapeutic doses over time, and we're actually able to somewhat replicate that same type of thinking in the testing process.
Right. Now, you were at AACR recently, weren't you?
Yeah.
And I unfortunately didn't get to go, but I know that organoids were all the rage at the conference.
Yeah.
So why was that?
Yeah, it was incredible to see. I think we had gone from a point of there being maybe a half dozen posters to essentially 3+ rows of posters
Right.
associated on organoids. So, and they were just jam-packed with interest in people. So it was very exciting to see. I think the answer is a bit multifaceted. We've had obviously an incredible number of failures in clinical trials, whether the reasons are around toxicities, unanticipated side effects, or simply they work in mice, but not in humans, right? As pharmaceutical companies, we've gotten very, very good at curing mice, lesser so at curing humans. But that's part one. Second really is around, I think, the cost of getting an IND submittal or to the point of an IND. It's become so expensive, so time consuming, and more data have been generated as more and more toys have been developed for folks, right? That requires more people, more analysis. You know, they're trying to make sure that by the time they get to that point, this thing is really going to work this time. And the reality is, you know, we're seeing partial improvements, but not historical improvements. And I think one of the other challenges historically has been, you know, when we finally do see something that works, we may not really understand what the mechanism of action is. I mean, there are a lot of drugs that have been approved and we don't know why they work, right. Which is, you know, a bit concerning sometimes, right? And then finally, you know, there is that big push to eliminate the utility or usage of animals as part of the process. And these in vivo technologies like tumoroids and organoids are proving themselves to be the best possible leap forward in drug validation technologies. And fortunately, here in the U.S., we had the FDA Modernization Act signed last year, which authorizes the FDA to accept these types of in vivo platforms as alternatives to animal studies. So we're really in that perfect juxtaposition of providing an enabling technology at the right time.
Brilliant. Bhuvanesh, would you like to add anything to what Dean has said, or has he covered it all so perfectly?
I think he has covered a lot of aspects of it, but it's from the time frame. I'll just add a small element to it. 5 years ago, when I started the company, I had to tell people what an organoid was.
Right.
Now come to the point where they're all rage in AACR. They had 3 ballrooms simultaneously talking about tumor microenvironment and organoids. And on Saturday, the first and most important day of the meeting. So I think it's becoming important, and people are realizing that they can use faster, better models, which are human on human context, for developing drugs rather than using mouse models. So that's where we are.
Fantastic. Now, who is the ideal client then for Spanios when you're talking about using your technology?
So I know this is a 2-part answer. I will answer from a scientific point of view. I,
Right.
as a mission-driven person, want to have a client who's interested in developing therapies for patients with difficult to treat cancers.
Right.
And I want to work with them so they're enabled to bring therapies into the market. That is my primary concern. Our goal is an efficacy first study is what we do. So the main goal is to figure out if the drug will actually work before we try and delve into the mechanism of action, before we try to develop all this peripherals that we need for IND filing. Our goal was the team that is interested in doing efficacy-based studies is the one that will be ideal for us to follow through. And that's what we try to do.
Right. And Dean, from your
Yeah, I'm glad you
perspective.
brought that up. I've been pushing efficacy first for many years. So having a model that can actually be achieved with is quite exciting. I would also like to add, I think we present a very crucial solution to a lot of the in silico pharma, like the AI machine learning companies that are developing computer-based structures and therapeutic libraries, but they lack, excuse me, they lack wet lab space or an ability to validate it. And you know, that's a much bigger challenge for these folks because there's a ton of them out there, and they're all trying to convince pharma that they've got the right in silico design molecule to move forward, but they don't have, you know, wet lab data to justify that. And having an efficacy first approach, you know, we're going to say, well, would you like to see what it does to a human, right? And being able to generate that type of data for these folks now enables companies like that, I think, to really move forward much quicker with just the follow-on stuff that's going to be necessary, the PK tox, the OTAs, the immunogenicity studies, all the things that are part of an IND. In the end, they're going to end up being able to keep more of their money when they're negotiating around the table with a Pfizer, Novartis, or whoever for the potential in licensure of their asset. So I think that's definitely one. I think number 2 is companies that are under massive cost and time crunch. Because the traditional approach is just a lot of reiterative testing, building cell lines, designing assays, having custom antibodies and probes made, buying tons of off-the-shelf kits, you know, tissue slides that you can have made or purchased, and it's just a lot of this, only to find out it doesn't work. And we're just like, we're of the mindset, test it on a human model first, before you waste all that time and money of the investors to move forward with something that ultimately you found doesn't work. With us, we can prove whether it is efficacious or not. And we'll give them the translatable data that they need to make those decisions.
Right. Now, one question I will ask you as well, though, is that collaboration and partnership has become a huge buzzword within life science and pharma, particularly since the pandemic. So, and I speak to a lot of companies, and I always say to them, you know, what do you see the future hold? And they all say, well, collaboration, we're always looking to look for new partners, look at how we can work. So from your perspective, are you open to partnerships? And if so, what would be the perfect partner for you?
Yeah, indeed. Definitely are. In fact, we expect to close on several very shortly, so stay tuned for that. We are also very open to support CROs as an example, these contract research organizations are supporting, big and small pharma alike with lots of capabilities. But the one thing that we've been able to master on these in the build out, maintenance, and testing of these tumoroids, very few companies can do. I mean, we're talking to big pharma, we're talking to larger CROs, and they've invested a lot of money to try to figure this out, and they have not been successful. So we're absolutely open to any sort of deal with a contract research organization and say, look, you have an opportunity and you want to be able to say, look, let's test on a human first, and then figure out what of the follow-on data is necessary, and then really enabling that CRO to tell their clients, yeah, we're going to move you through faster the process to an IND. That's an exciting place to be in for certain. Yeah.
Brilliant. Bhuvanesh, anything to add to what Dean just said?
Well, in terms of partnerships, there are always different aspects to it. So what he has talked about is the commercial end of the partnerships. But as we develop better models, we're developing academic partnerships and industry academia partnerships to further the field so patients get help. And that's the primary concern we have as a team, is eventually we want drugs coming to the market faster. And in order to do so, our goal is to enable whoever is willing to partner with us. And we have partnerships with some academic institutions already in play to build out the next generation of models, to build out other things like that. So we are all working together with other companies to get these things working.
And that leads nicely actually to my final question, which is, what do you see the future hold for Spanios? And actually, I suppose, what do you see the future hold for technology around organoids and tumoroids? Because obviously you talk about, and you mentioned AI just a bit earlier as well in terms of AI companies. So how do you see that developing going forward?
So there are two elements to it, right? The first part is what future holds for us in terms of what we are building. We always propose to build a model that is more representative of the human condition. We also want to ensure our models are well characterized. If they're well characterized, they are more easy to utilize for developing preclinical studies. So those are the immediate future needs. In terms of long-term for the tech, organoids are here to stay. Whether they be tumoroids, whether they be organoids, they're here to stay because it's easier, more human context, it's more relevant in terms of studies to do. I, being a person who worked on mouse or animal models for several years before I did this, can tell you they are better in defining what a humanized system looks like. And it is the whole idea. You want to be able to test an as close a system as possible. Cost is an important factor. Eventually, more than cost is the time. You do a year and a half long study before you realize the drug's working or not working. It is not a very efficient way of moving drugs to the market. Our models currently are enabling already, studies which are new drugs, repositioned drugs. They're okay in some other kind of disease, they can be repositioned for this. And the last bit is to take multiple fancy new immuno-oncology drugs that are coming in and combinations thereof. Those need to be addressed. And I think organoids will be the ones, and tumoroids will be the ones that will stay out of them.
Yeah, right. I think I might add a little bit to that also, in that we are agnostic as an organization. If you've got a solid tumor you're going after, we are pretty certain we can build organoids to that solid tumor cancer indication. Sure. But we're also therapeutic modality agnostic. So whether you're working on a small molecule, a large molecule, a VHH antibody, a peptide, a CAR T, a CAR NK, a CAR MAC, you're doing TCR based or similar type of work, or synthetic lethalities even. We can test those out with our platforms. We have a number of platforms specifically designed for each one of those types of therapeutic modalities. So we're quite excited about having that flexibility to support all solid tumor research pharma, regardless of the modality type. And we're in the position to do it for certain.
Fantastic. Well, look, gentlemen, I appreciate taking time out to talk to me today. It's a really interesting topic, and really delighted we've had the chance to talk about this, because I've learned something new, certainly, in this conversation. Now, if people would like to know more, though, and I know you're on the road as well, Dean, at a lot of events as well,
Yes.
as is the company, where can people meet you? Where can they get more information?
Sure. Definitely at events like AACR, ASCO, various ESMO events, we tend to attend those, but definitely go to our website, which is just simply Spanios, Spanios lab.com. And there's a contact us link on that page. And you could always drop me an email or reach out via LinkedIn. My email is simply deanremy@spanioslab.com. Happy to answer any questions that you might have, provide information, or provide a scientific deep dive with our science team, including Bhuvanesh here. So yeah, we're really excited about the response that we've had. We've been enjoying conversations with most of the big pharma and literally dozens and dozens of the small to mid-sized guys. So all this since really the start of the year.
Brilliant. Well, look,
Yeah.
obviously, like all these things, timing is everything. It looks like you're at
Yes.
the right time in the right place.
Yeah.
So I
Yeah.
wish you lots of success with the technology. So Bhuvanesh, Dean, thank you very much for taking the time out to talk
Thank
today.
you.
It's been really interesting.
Thank you so much for your time today, Ruslan. Always great to reconnect.
Thank you, Rizwan. Thank you again.
Thank you. And viewers, so there you go. Hopefully that's whetted your appetite and you'd like to learn more. So if you'd like to know more, then firstly, obviously, you can check out the website. But secondly, as Dean said, you can meet them at numerous events, which will be no doubt listed on the website as well. But you can contact them directly via LinkedIn as well. And if you've got any questions based on what was discussed today, feel free to put them in the comments below. It would be great to hear what you think about the technology around organoids and tumoroids and how you see those models work in comparison to the more traditional models as well. So thank you very much, gentlemen. Viewers, thank you for watching. And until next time, as always, stay well and stay safe. Bye-bye.
Bye-bye. Thank you.